What's going on? This is Deanza. We are going over eukaryotic regulation. Got a lot of slides on this one to get through, so I'm not going to waste any time. Let's get rolling here. There we go. Here are the learning targets. Describe ways that eukaryotes control transcription and translation, and describe epigenetics. And a lot of times you see this pre-transcriptional control or post-transcriptional control. Those are some terms we should be familiar with. So let's get started. First, let's talk about a virus. A virus is something that needs a host to survive, and we've been learning a lot about viruses over the last couple of years here. With a virus, they have a protein coat and they have some kind of nucleic acid. The virus injects its nucleic acid into the host, and that creates a virus factory. That genetic material that the virus inserts gets transcribed and translated into proteins to make a new protein coat, and the genetic code gets replicated to go in there, and that means that the host cell all of a sudden becomes a little virus factory and starts making up viral particles. Viral genomes can be DNA, can be RNA, just depends on the virus. Every virus is going to be a little bit different. When we look at that, they inject a material, will take over and make a little things called virions or virions. I usually say virions, I think. And those are just little replicas of the original virus that came in. When the virus does that, that will, and enough viruses are created, basically that host cell dies. And as that host cell dies, it goes through what's called the lytic phase, where during the lytic phase, the host cell is actually going to rupture and release all the bacteria. So let's look at this picture right here. When we look at our virus, and this is a T4 bacteriophage, this is showing a bacterium right here, T4 bacteriophage right there. It inserts its viral genome. That viral genome has a promoter. You should know what that is. That's where transcription starts. And we have our gene sequence. We have other genes, another promoter, more gene sequence. And this first segment codes for, well, we got some genetic material copied here, and we have a enzyme to cause more transcription going on here, kind of something that's going to assist and tell the RNA polymerase what to do, make some more stuff. Now we got a viral body, and we got an enzyme for lysis. Lysis means that that's going to cause that bacterium to burst. That's when it tells the bacteria, okay, you're dead, and you're going to release our little viruses out there. Well, that's the basic pathway that things work with, where you have a promoter, we have genes, they get transcribed, that transcription, DNA to mRNA, mRNA to proteins. Sometimes the proteins tell us, hey, let's make copies of the genome, whatever needs to happen. This protein is going to go to this gene, and that's going to help transcription happen there, turn into more enzymes or proteins over here, and then eventually we have this lytic phase right here. So that's a basic way that prokaryotes are infected with viruses. Well, eukaryotes are very similar to that. Eukaryotes have negative controls, which turn transcription translation off, and positive controls, which turn transcription translation on. Anything that binds to a promoter and tells us, hey, we're going to start transcription, we just call them transcription factors. Could be probably going to be some kind of protein that's in the cell somewhere that's going to bind in the area that's going to tell the cell, hey, we need to get this going. With eukaryotes, usually there's a few promoters that are going to take place. Sometimes the DNA folds over on top of itself because there's promoters somewhere, transcription factors somewhere else on the DNA that need to get over to that area, and that all signifies that, okay, it's time for transcription to happen. In eukaryotes, there's a promoter, just like in the prokaryotes, and there's a specific area in the promoter called a Tata box, and depending on who you hear it from, they may say Tata box, Tata box, I say Tata box, and it's just an area in the promoter that has a bunch of thymines and adenines. So we have TA, 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 TA. Well, TF is transcription factor, and we have transcription factors that cruise in, all of these other molecules that attach through. Once everything gets there that's supposed to be there, then the RNA polymerase can transcribe that segment of DNA. And it's all because that Tata box is open and available and allows the transcription factors to, those basically tell the products that are transcription factors where to bind on that genome. Now, we've talked about every cell in your body through mitosis has an exact copy, an exact replica of all the other DNA. So all the cells have the same DNA, but cells do different things. Well, when cells are doing different things with the same DNA, we say that those cells have been differentiated. They have gone through a process where now they have a specific job. And realistically, in a cell, a lot of times cells will start out as stem cells. Stem cells are blanks that don't have a purpose yet. They get a specific signal that tells them, okay, you are going to be a skin cell. You're going to be a kidney cell or a liver cell, whatever the cell is going to be. And then it only uses the genes that are needed for that specific cell and tissue. I don't want my kidneys doing liver stuff. I don't want my liver doing kidney stuff. So we have all of this stuff going on, and these transcription factors are going to make a lot of this stuff happen. And controlling transcription and translation play a really big role in this because, well, you got 20,000 genes. You don't need all of them for every cell. Certain cells need certain things, and as those things are turned on or off, that allows the cell to do what it's supposed to do. So when we look at regulation, well, promoters for eukaryotes, those are going to be diverse. They're not all going to be similar. Well, bacteria is pretty basic. Eukaryotes are a little bit more complex. And like I said, there might be more than one promoter in a gene, and there also is going to be that thing called the Tata box, where that's going to be the area for the transcription factors to attach to. General transcription factors are things that are going to bind to that promoter. Positive ones are going to activate transcription. Negative ones are going to repress transcription. Same thing that we talked about with the prokaryotes. Repressing means you turn the gene off. Activating means you turn the gene on. So a lot of times that happens with repressors that may attach and block transcription, period. Positive things are going to turn transcription on. They're going to make the cell make RNA. Negative ones are going to turn it off, so RNA can't happen at this point in time. Now, when we look at this, there are other things called enhancers that will bind to those activators. They are going to assist with turning genes on. Sometimes there's things called silencers that will bind to repressors, and that will turn genes off. And again, super sciencey names. If it silences it, it shuts it off. If it enhances it, it turns it on. This all has to do with positive, negative regulation. Multiple transcription factors are usually going to be involved when we're talking about eukaryotes and transcription translation. A lot of times I mentioned earlier that other segments of DNA, all the transcription factors might attach far away from the gene, but the DNA bends and folds on itself and will allow it to turn on. So there might be different spots along the gene where a bacterium, all of the genetic information is always together because they only have one circular chromosome. Eukaryotes, in humans, we have 23 pairs of linear chromosomes. So there might actually be places in varied spots along the DNA that cause transcription to occur. So the DNA may move around and allow that to happen. Sometimes transcription is changed based on what is going on around the organism and what's going on around you. Your environment actually plays a large role in that. When we look at environmental factors, they can cause stress. And stress, oh, you gotta test an AP Bio, that's stressful. Okay, let's talk about stress. Yes, that is stressful. I'll agree with you there. But let's talk about stress from a biological, physiological standpoint. You haven't eaten in 3 weeks. That is stressful. That's going to change how your genes are working inside of you. In plant cases, maybe it hasn't rained in a while and there's a drought. Well, that changes how the plant does protein synthesis, and it modifies what it does based off environmental stresses that happen around it. When we talk about environmental stresses and things changing our genome and how things are transcribed and translated, we call that epigenetics. Epigenetics has to do with not all your DNA always works the same way. It changes based on things going on around you, whether it's the environmental stresses around you, the food you put in your body, the things going on in your life, whatever may be the case. DNA is malleable. It's not a solid static thing. It sometimes has some variation into it based on what's happening around us. Now, a mutation is a permanent change. Mutations are not reversible. Epigenetics is, and we'll talk about how epigenetics happens in these next few slides. One way it takes place is through the use of histones. Now, we talked about, a long time ago, we talked about DNA being long, stringy stuff called chromatin. Well, that chromatin is super duper long. You get about 3 meters of DNA inside of each cell, that's about 10 feet almost of DNA inside each individual cell. Well, how do we fit all that stuff inside a cell? We use these things called histones. Histones are little protein complexes that the DNA winds around like thread on a spool, and that really condenses the DNA. When we talk about a histone with the DNA wrapped around it, that is called a nucleosome. Well, if you look at these histones, they have these little arms or tails that come off of them. Sometimes those tails actually move where they actually grab and hold the DNA really tight. Sometimes they open up and allow the DNA to uncoil. So depending on what's going on around you, your DNA may be wrapped up around this little spool. And if there's a gene that, in this case, is sitting right here and here, that's not going to be able to be transcribed like a gene that's from here to here in this picture. So if a gene is wrapped up in here, it's not going to get transcribed. If a gene's in an exposed area, it will get transcribed. So these histones play a large role in changing what can happen inside the cell with regards to transcription and translation. If the DNA doesn't get transcribed, then there's no translation, no protein is made. If it does, then yeah, the protein is created. So, how do histones do that? Well, if you add histone acetyltransferase, histone acetyltransferase, what do you think that is? Yeah, it's an enzyme, and it's histone acetyltransferase. Let's go back here. Let's look at our picture really quick. We got our histone, we got our arms, all of that good stuff, is going to neutralize the charge of the histone. When that happens, the histone, we say, is acetylated. When the histone is acetylated, that means the tails are going to let go of the DNA, and the DNA is going to unwind off of that spool. Well, do you think that's going to be a positive or a negative factor? If all of a sudden there's more DNA available. Yeah, positive. Very good, very good, very good. Let's go to the next one. Well, we also have negative factors that can occur. Histone deacetylase. Remember we said the acetyl groups make the histone open up and let the DNA unspool? Well, histone deacetylase is going to take away those acetyl groups and cause those tails to grab on tighter to the DNA and make it so that they're not as available. So that's going to be a negative factor. Another thing that can happen to DNA is methylation. Methylation happens when, guess what? Methyl groups, super sciencey, get added to DNA. Well, when adding methyl groups, that could be a positive change, it could be a negative change. I usually associate it with a negative change where it's going to sort of shut some stuff down, but it's also with signaling and all of that. Same thing with phosphorylation. Adding phosphate groups happens a lot when the chromosome condenses. Well, when do the chromosomes condense in the cell cycle? Yeah, of course, it's prophase. Prophase of mitosis and prophase of meiosis. The chromosomes are going to condense. There's not much protein synthesis going on during that time. When the chromosomes condense, that's to cause a cell to divide. So phosphorylation is going to be a very negative factor when it comes to the transcription translation thing, because it's going to sort of tighten the DNA up so that it can go through another process that needs to take place. And all of these things, which are nice, are reversible. So we can methylate things, we can take methyl groups away. We can phosphorylate DNA, we can take phosphate groups away. Excuse me, I'm struggling over here. You can grab onto the DNA a little tighter, you can let go of the DNA. So that's very different than a mutation, which is a permanent change to the DNA. And since this is happening in DNA, these are all pre-transcription factors that are going to limit or enhance transcription and translation. Look here, we got our DNA with a bunch of histones wrapped around. When histone acetyltransferase gets added, that's what these little red dots are being called. We're transferring acetyl groups onto the histone. It's the enzyme that does that. That's going to open up the DNA strand now and allow for remodeling protein. We got some transcription factors and some transcription complex, and it's going to go transcribe and then transcribe the DNA into RNA, and then the RNA will get translated into protein. So this area got opened up, and then after it's done, add histone deacetylase, and it tightens everything back up, and the DNA is wrapped up nice and ready to roll. When we talk about DNA methylation, usually it's going to be a cytosine base, and these methylated DNA proteins tend to repress transcription. Like I said, I usually think of this as a negative factor when I think of methylation. This is just something that we see, we call them CPG islands. This is areas where you have a lot of methylated cytosine that is present in the DNA at that one point. More methylation. Enzyme called demethylase removes the methyl groups. Demethylase, ACE enzyme, demethyl, you take it away. So again, something that can go backwards and forwards when we're looking at the overall picture of this. One more, there we go. All right, so when we are looking at chromatin, remember chromatin is a long stringy DNA. Euchromatin is going to stain lightly. That one's usually going to be transcribed. This is a chromatin that basically has opened up and allowed the DNA segments to be open to transcription, which will eventually lead to translation. Heterochromatin is going to be a little bit darker. That happens when chromatin is methylated. When there's a lot of methylation, that blocks transcription. Now, we talked about males having the X and the Y chromosome, and the Y really doesn't have very much information. And I said, don't worry, ladies, you'll get your chance when we come talking about methylation. Well, women are like, oh, we got 2 X chromosomes. They have a bunch of genes on them. Blah, 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 blah. Ha-ha. One of those X chromosomes is all methylated. So it really doesn't do much. There's no transcription or translation happening there. That's something that we call a bar body, B-A-R-R body. That's just a name for it. I'm guessing Dr. Barr found those. And yeah, it's because you only need 1 X chromosome doing its thing inside each of your cells. We do need the right number of... Now, that being said, you can't just have female DNA come together to make a child. You need the female and the male because it has to have the right number of transcribing active genes. Too many transcribing active genes causes issues, and if you put... Yeah, it just messes stuff up. Read that section of the book, goes into it a little bit over there. Almost there. 4 more slides only. 4 more slides. Sorry, this one's a little bit longer one. With the environment, we've seen this, these issues and these changes that are reversible in identical twins, looking at studies of them. This has to do with the epigenetics. Identical twins have identical genomes, and we have seen that the environment does absolutely play a role. And just think about things that you put on in your body from the environment that can absolutely change things. We know tobacco smoke's god awful for you. We know diet's super important to how we work, and we know stress happens to us all the time, whether it be psychological, emotional, or physical stress that takes place. All of those things cause different changes in sets of twins, leading to the idea that, yeah, your environment does absolutely matter in the way that you grow up and the proteins that your cells express. There we go. So this is sort of what I was alluding to earlier. Methylation is going to differ between the eggs and the sperm, and that's why, women, you can't just do away with us. You can't just take your DNA and make a super baby with only female DNA. Nope, you need the male imprintation. You need the male methylation patterns as well. It does something called genomic imprinting, where you put specific information into the growing child, and they have to have chromosomes from each. The methylation pattern is required from the male and the female to develop a viable embryo. Oh, I completely lied. We don't have 4 more slides. We have like 8 more slides. Sorry, this one's going to go a little over the 20 minutes. Sorry about that. All right, post-transcription. Now, we were just talking about stuff pre-transcription. Post-transcription is controls that happen after transcription happens. So pre-transcriptional factors limit whether mRNA can be made. Post-transcriptional regulation happens after the mRNA was created. Well, one of them we've talked about before, splicing. Our mRNA gets spliced. Well, depending on how that mRNA is spliced, we can get different proteins. We call that alternative splicing. When we look at this, exon 1, intron, exon, intron, exon, intron, exon, intron, exon, intron, exon. Well, we know, pardon me, that we transcribe DNA to pre-mRNA, it's an exact copy. And then we're going to splice out all those introns, but depending on how we splice from here, we're going to get different proteins coming in. This uses numbers 1, 2, 4, 5, 6, 1, 3, 5, 6, 1, 3, 4, 5, 6. So depending on the combination of portions here that are going to eventually get translated, we can make 3 completely different proteins all from the same gene. And that's what alternative splicing is all about and how that works. MicroRNA is, sorry about that. MicroRNA is something that will inhibit translation. This is the stuff that's going to chomp up the mRNA after it's been used. So microRNA is going to come in and it's going to chomp things up. Remember we talked about adding a poly A tail to the pre-mRNA transcript as one of the modifications in the last chapter. Well, this is one of those reasons we do it, because the microRNA wants to chomp up that mRNA molecule, and the poly A tail sort of keeps it going until it doesn't need it anymore. MicroRNA, we talked about, it attaches inhibits. We can modify the GCAP. If the GCAP is unmodified, it does not translate. Modified GCAP will translate. So depending on the GCAP that's put on and modifying the GCAP after it's on will determine whether or not that mRNA can bind with a ribosome. There are also translational repressors. Translational repressors work just like transcriptional repressors in that they bind to mRNA instead of DNA. When they bind to the mRNA, that's like blocking it from binding to a ribosome. So different ways that we can shut off the mRNA, similar to how we shut off the DNA before in the last section. This was a picture from the book just showing that microRNA is going to chomp it up and go through things. Precursor RNA folds back on itself, forming a double-stranded mRNA. Dicer protein cuts the RNA into small fragments. MicroRNA comes in, chomps it up, and leaves us with a bunch of free bases. Now this is post-translation. Proteins get broken down. When proteins are no longer needed or used, they go through a process where they get digested. Ubiquitin is something that's going to attach to the protein, and it's going to bring the protein to something called the proteasome. Proteasome is another protein, no surprise there, that is going to act like a garbage disposal and break up the ubiquitin attached protein sequence that is all done. And when it breaks it up into smaller polypeptides, or it can break it up into individual amino acids. All of that stuff can be recycled within the cell. So, when we look at the picture, this makes a little bit more sense. Here's our protein. We don't really need it anymore. Ubiquitin is a chemical inside our cell. It's going to attach to that protein. It's going to bring that thing to the proteasome, which to me, for some reason, looks like a garbage disposal. Look underneath your sink if you have a garbage disposal. It looks kind of like this, actually. That protein gets fed through the proteasome, and then it gets chomped up into smaller individual units and individual pieces. So, we talked about a lot of different ways to mess around with transcription in eukaryotes. We talked about ways to mess with transcription. We talked about ways to mess with the mRNA, mess with translation, and then we even just mentioned how to mess with the protein once we're all done with that. Well, your book gives an example of viruses in the eukaryotes and how they work. Different ones are going to work different ways. This is a term you should absolutely know, a retrovirus. Retrovirus is HIV is an example of one, and that's one that is going to, let's go here to this next one. There we go. It releases something called reverse transcriptase. Well, that's an enzyme. Reverse transcriptase makes a DNA strand that is complementary to RNA that's in the HIV. So HIV actually provides RNA, but reverse transcriptase reads the RNA to make a DNA strand. That DNA strand gets lodged into the host DNA segment, and that creates what's called a provirus. A provirus is the viral DNA that's hiding inside your genome. That HIV DNA came from the HIV RNA. The reverse transcriptase turned the RNA from the HIV into DNA. The DNA incorporated itself into your DNA and becomes a provirus, and eventually that provirus lays dormant and eventually can transcribe and translate viral DNA, viral RNA, viral proteins, which, same thing with the bacterial one that we talked about before, creates all new little baby viruses, which is one of the reasons HIV is not a good virus to have. When we see this here, here's our single-stranded viral RNA with the reverse transcriptase on it. That's what that yellow dot is. This is the HIV virus itself. It attaches to our cell, releases its DNA, its RNA, That RNA is going to do reverse transcriptase to make viral DNA. That viral DNA incorporates into your DNA. It's called a provirus at this point because it just sort of lays there, and at some point throughout your life, at some point, that viral DNA that's staying in your cell will get transcribed and translated, creating more little viruses, and they release them into your body. And you spread them to your body and potentially other people, which is one of the reasons HIV is no good. There's a good short about that in the book towards the end of the chapter. Please make sure you go check that out. A lot of information in this set of slides, a lot longer than I wanted it to be, but we just sort of knocked it out and got it going. If you have any questions, please reach out. Otherwise, check your learning targets, and you have a good afternoon.